Um, okay, well, I'm going to be speaking to you today about black holes and event horizon. Oh, wait, you're a biology group, not a physics group, right? Um, well, actually, I'll tell you what. This is actually the nanoverse, not the larger universe. So we'll come to what this slide is in a minute. I'm going to be talking to you actually about the coordination of plant cell division and differentiation. Um, these things here that you see are actually nuclei stained with dye that fluoresces blue when you shine UV light on it. And as Bill said, my name is John Larkin from Louisiana State University, here in our biological sciences department. First thing, I want to remind you that for anybody who might be interested in graduate school, not just at LSU, but particularly in our biological sciences program, there will be a free pizza lunch next Tuesday after this session next Tuesday from 11:30 to 11:30 in the morning till 1 p.m. in room 28 in the Life Sciences building, which is in the old part of the building and down in the basement. We have a rare thing in Louisiana, a basement. It occasionally fills up with water, but not too often. And there will be a very brief presentation, like 3 or 4 slides. Why go to graduate school? Why go to graduate school at LSU? What you can do with a graduate degree, and a little bit about our graduate program and how it's organized. And there will be graduate students and faculty there to answer your questions, so you get to talk to real live graduate students as well as just faculty. We're trying to give you a chance to talk to the grad students, not just us. And also, if that sounds boring, well, there is the free pizza. Anyway, and there's a sign-up sheet that will be available afterwards, so please sign up. I think it's over there somewhere. And the only reason to sign up, you can come if you forgot to sign up. In fact, anybody who's in these other places, if you want to come drive down here and join our free pizza lunch, you're welcome to join us too. We'll post the slides and the recruiting information online. There'll be some flyers available on our website and some other things. Okay, so that's some business. Let's get on to the biology. Well, I work on a plant. And what I'm going to try and persuade you is that some of the things I work on with this plant might even be a little bit relevant to humans. So plants aren't totally boring. The other thing is, actually, when you think about health, well, doctors have a lot to do with health, but if you read the news or think about things, what you eat has a lot to do with health too, and a lot of what we eat is plants. So studying plant biology can be related to health and directly related to human concerns. I work on a plant that is a little tiny thing. It looks big on this screen, but really the plants are about this tall. The seedlings are about the size of a pencil eraser. But this is the plant Arabidopsis thaliana, which is kind of the botanical fruit fly. Just like we study fruit flies because they're easy to grow in the lab, you can grow lots of them, and it's easy to do genetics and molecular biology with them. This plant is kind of the lab rat or the fruit fly of the plant kingdom. So scientists love it. It's not particularly good for anything. It's related to mustard greens and broccoli and cabbage. But when you look at these leaves, well, that's a leaf, and you can see these little white kind of star-shaped or cross-shaped things on the leaves. These are leaf hairs, which are, the technical name is trichome, from the Greek word trichos, which means hair in Greek. You probably maybe know some medical, anatomy terms, even in animals, that have that same root where you have cells that are hair-like. And we take a closer look at these hairs. This weird branched structure here with the little dots that are kind of crystalline incrustations on a really thick cellulose wall that's really tough and rigid is a single cell. And these cells, you notice they're sharp and pointy on the tip. One of their main functions is that they act as kind of microscopic barbed wire to keep insect larvae that might want to chew on this plant leaf from thinking it's quite as tasty because they get a mouthful of prickers if they do. Well, what interested me when I was doing my postdoctoral work was that this cell is large, it's strangely shaped. Here's an ordinary flat epidermal cell. Well, the phenotype of this cell and the phenotype of this cell are radically different from each other. And that was what interested me at first, and we were interested in how do cells become different from each other. Well, I'm not going to tell you anything about that much, even though that's where I started, because we made a kind of shocking discovery, or shocking to us anyway, really interesting discovery, that suggested that part of this developmental program that made this cell so large involved control of the modifying the cell cycle, so instead of going through mitosis, the cells did something a little different. So now back to our black hole slide. You remember I told you that each of these is a nucleus stained with a DNA binding dye that fluoresces when you shine UV light on it. So the blue fluorescence is the nucleus. Now let's go back. You'll notice that there are some individual cells arranged around the base of the leaf, and they're raised up a little bit. Each one of those cells has a nucleus, and then the nucleus of this cell is up about here. If you imagine this leaf, and I'm going to drop a cover slip probably as big as the front wall here on this poor leaf. This is a scanning electron micrograph picture, but there we go. And if you imagine this hair, this trichome leans over like this, so the nucleus is going to be a little off center, and there's a ring of nuclei there. Well, here we have the ring of nuclei of those support cells around the base of the trichome, and here's the nucleus of the trichome. Do you notice anything different between, say, this nucleus and this nucleus, besides that this one's a little bit out of focus? It's huge, okay? So these nuclei are different in size. And the thing that's interesting about this, so this one compared to this one's a lot bigger. And this dye binds roughly in proportion to the amount of DNA that's there. So that says there's more DNA in this nucleus than in this one. But wait a minute. Aren't all cells in a eukaryotic organism, in a diploid eukaryotic organism, shouldn't they all have 2 copies of every gene? Well, it turns out that that's what you mostly learn in textbooks, but it's not entirely true. Many plant and animal cells have more than the diploid DNA content of DNA through a process called endo replication, where after the cell stops dividing, it keeps replicating its DNA. And typically, this is giant cells. Very large cells have this kind of process. In the case of trichomes, this nucleus probably has 32 copies of the DNA, not the diploid 2 copies of the haploid genome. So it's got 32 copies of the genome. In fact, these cells probably aren't diploid. They probably have about 4 copies of the genome in them. It turns out this is very, very common in a wide variety of organisms. I want to show you just a couple of examples to show you why this might be important. Here's one example that we eat. This is a tomato that's thought to be closely related to the wild ancestor of the cultivated tomato, Solanum pimpinifolium. It has a tiny little cherry tomato fruit. This is our edible cultivated tomato. It's a lot bigger, just like that nucleus was a lot bigger, right? Well, it turns out that about half of the increase in size between here and here is enlarged cells with endo-replicated nuclei. The other half of the difference is an increase in number of cells caused by a longer period of cell division. But some of this difference is due to giant cells with endo-replicated nuclei. And if we want to look a little closer to home at human biology, this is a human embryo at the point of implantation. All of the green and blue cells are derived from the fertilized egg. The green is the embryo proper and will develop into the baby. The rest of this tissue is all support tissue coming from the egg. And here we have the uterine wall. And you can see that this blue layer of cells, where it contacts the mother's tissue, these cells are enlarging and burrowing into the mother's tissue. Eventually, this whole embryo will be implanted in the mother's tissue, and a layer of maternal tissue will wrap around the outside. But these cells, the trophoblast cells, endoreplicate their DNA up to 1,000 times the haploid content. So 500 times more DNA than a diploid cell. And these cells actually play a major role in attracting blood vessels from the mother to give a nutrient supply to the embryo, and a major role in transfer of nutrients to the embryo. These cells, and this is their endoreplication and extending processes into the maternal tissue as giant cells, is absolutely essential to implantation and survival of the embryo. So this process of endoreplication shows up over and over again in giant cells, metabolically active cells. We don't 100% know why, but we're working on that. So how can this happen? Well, here's the cell cycle, and most of you, since you're all biologists interested enough to spend a summer doing research, probably are familiar with the cell cycle. The G1 or gap or growth 1 phase, the longest phase of the cell cycle, an interphase, where the cell is growing and preparing for DNA replication, which occurs in S phase. G2, the second gap phase, where the cell is preparing for mitosis. Well, these stages of the cell cycle are controlled by enzymes called cyclin-dependent kinases. Here we have one set of cyclin-dependent kinases that trigger S phase at the end of G1, and a second set that trigger mitosis. This is really an oversimplification, but it'll do for our purposes. So what is a cyclin-dependent kinase? Well, you probably know that if you stick the ACE ending, ASE, on the end of a protein, that usually means it's what kind of a protein with what kind of a function. It's some kind of an enzyme, right? Well, a kinase is a special kind of an enzyme that puts phosphate groups, takes a phosphate from ATP and puts it onto another protein. So let's take a look at how that might work. Here we have a mitotic CDK, or cyclin-dependent kinase. We have ATP, and we have a target protein, histone H1. Does anybody know where in the cell histones are found? In the nucleus, on what kind of structures in the nucleus? Chromosomes, so chromatin. So this is a protein that's on chromosomes. And if you're going to go through mitosis, the cell needs to move those chromosomes. Not only does it duplicate them, but it needs to move them to opposite ends of the cell with the mitotic spindle, right? So at the end of the summer, when you move to a new apartment, maybe, what do you do with all your stuff? You got your stuff all spread over your room, and then when you go to move, you don't move each individual thing, right? You pack it up into boxes. You condense your stuff before you move it. Well, the cell's got to do the same thing. The chromosomes have to condense. So how does it do that? Well, first of all, this cyclin-dependent kinase is called cyclin-dependent, so it must be dependent on a cyclin, which is another kind of protein. And then when it binds its cyclin partner, which only is synthesized in G2 just before mitosis, then it can convert ATP to ADP, and the phosphate group gets transferred to particular amino acids on the histone, serines or threonines, that have a hydroxyl group. And the end result is the chromatin condenses, the chromosomes get packed up and ready to move. There are actually hundreds, if not thousands, of target proteins in the cell that get phosphorylated by cyclin-dependent kinases at different stages. But this is a protein kinase, and the word kinase actually is, if you ever see it, tells you that phosphate groups, these are enzymes that put phosphate groups on proteins. And so cyclin CDK complexes phosphorylate target proteins and change their activities, triggering events in the cell cycle. And this is only one of many, many events that gets triggered by this kind of reaction, just as an example. OK, so if endoreplication involves replicating the DNA and skipping mitosis, and we've got kind of two flavors of cyclin-dependent kinases, some involved in triggering S phase and some involved in triggering mitosis, it stands to reason that we have to keep these ones active and going in the cell cycle, and we got to get rid of these. So endoreplication involves skipping this phase of the cell cycle and eliminating G2 in mitosis, so we just get alternating S phases and G1 phases. I keep wanting to look at all of you. I don't want to forget all of you in the other rooms around the state. I keep looking at the screen and remembering that when I use Skype, you always want to look at the screen, and really the person you're talking to is inside that camera there. I don't know where the camera is, but that's all right. So hi to the audience at home. I don't want to forget you're there. So anyway, we can call this an endocycle instead of a normal cell cycle, the endoreplication cell cycle. So it's a modified cell cycle. It still keeps cycling, replicating the DNA, but we skip mitosis. So let's go back and look at these trichomes. Remember, this is a giant single cell with an enlarged nucleus. That means it replicated its DNA several times. A diploid cell has 2 copies. One extra round of replication gives 4 copies. Another extra round gives 8 copies. Another extra round gives 16 copies. Another extra round of replication gives 32 copies. Whereas this cell over here might be diploid or might just have 4 copies of the DNA. And along the way, the cell keeps growing and becomes this giant structure. And some other events are going on that cause it to be large and branched and crazy like that. OK, well, we found one of the approaches my lab uses is we find mutants. We start from mutations that cause some interesting phenotype, and we were looking for mutants that affected development of these leaf hairs. And we found a mutant. Now, a mutant is like if you break something, if you walk into an automobile, if you wanted to know how automobiles were made and you couldn't figure out how this happened, and you walked in sort of blindly and walked up to a worker and tied his hands behind his back or took him out of the shop and then looked at the automobiles and said, what goes wrong? When I tie that guy's hands behind his back, the automobiles come out with no steering wheel. If I tie this guy's hands behind his back, there's no doors on the car. If I break this robot over here, things don't get welded together. You know? So we can't see inside the machine so well, the machine being the cell, but we can break stuff. And if we break stuff selectively, we can say, oh, there's the part that I broke, and when I break that part, this fails to happen. Now, you need biochemistry to tell you how to put it back together, but the problem is biochemistry is not good enough to take all the pieces and put a whole cell back together and make it come to life, right? So you kind of got to work from both ends. So genetics works from the big picture. We break some stuff, find a single gene that's, when it's mutant, something goes wrong. And we found a really cool gene. We treated plants with a DNA damaging chemical and found a mutant that behaves just like Mendel said it would. It's recessive to the wild type. We can cross it, and it gives you all that nice Punnett square kind of stuff, 3-to-1 ratios and all that cool stuff. And typically, you just learn to memorize that stuff. But how you actually use it is this trick of you break stuff. You say, oh, when this gene's broken, something cool happens. And what cool happened is that we noticed, again, here's an electron microscope picture, a scanning electron microscope picture. You can see there are kind of some creases or segments here where it looks like this is one piece, this arm coming out here is one piece, and laid across the top of it is another piece. You can see the shape's a little bit funny, but it still looks pretty much like one of those branched leaf hairs. Well, when we cut thin sections through these and looked at them through the light microscope, what we found was that instead of being single cells, here, these were multiple cells. 1, 2 here, 1, 2, 3 here, sometimes up to 6 or 8 cells in a single hair. So if you knock out, we called this gene Siamese because sometimes you saw two hairs that kind of split, looked like two hairs joined together like Siamese twins, SIM for short. So these mutant trichomes are multicellular, and the amount of DNA in each nucleus is reduced. So what seems to be happening is that in these, if we tie this gene's hands behind its back and eliminate that function, these cells divide when they were supposed to endoreplicate. So that says that the function of the normal Siamese gene is what? Keep them from dividing, right? It's to make them endoreplicate, make them keep replicating their DNA and not divide. This is what happens when the function is missing. So what happens if we do the opposite? We engineered a plant to make too much of this Siamese protein. When you don't have that function, cells that are supposed to divide, that are not supposed to divide, divide. By the way, if cells that aren't supposed to divide start dividing, what kind of disease is that called in humans? Cancer. So right away we thought, this is interesting. We should study this, and maybe somebody will even give us money to keep studying it. And they have for the last dozen years or so. Well, but we wanted to know what happens in the opposite case. If we lose the function, the cells divide when they're not supposed to, what would we predict to happen if we make too much of this protein in cells that are supposed to keep dividing? What do you predict? Should stop dividing, right? He said stunted growth, and this looks pretty much like stunted growth, doesn't it? So that's kind of cool. Well, I guess the title was up there, but still, it gets cooler than that. Here is a wild-type epidermis in the EM. There's a trichome, but notice the scale bar. That's 200 microns. And right there is an ordinary epidermal cell. If we look at those stunted leaves, what we see instead are giant cells. And here again is a 200 micron scale bar, the same size. Now we've taken little bitty cells, and by forcing them to make too much of this SIM protein, we get giant cells. And if we look at the DNA content, on this axis we've got DNA content. Here's wild type. The normal epidermal cells have a very low DNA content, 2, 4, or 8 copies of the DNA. If we look at these giant cells, they have 32, 64, even 128 copies of the DNA. And just to convince you of that, here are the nuclei stained for DNA content. Here are some ordinary epidermal cell nuclei in wild type. And here are some of these giant nuclei in plants engineered to overexpress the genes. So Siamese, if we make too much of it, we can force cells to endoreplicate that weren't going to endoreplicate. So we found a switch, something that controls the process. Now I want to very quickly tell you what we're up to lately. So just to summarize, Siamese appears to, its normal function appears to be to suppress mitosis and turn on endoreplication. Does that make sense? You with me? We know, it turns out that after, it took about 4 years of work, but we found that the SIM gene encodes a small protein that resembles a few proteins that are known to inhibit cyclin-dependent kinases in other organisms, and all land plants. That's everything down to mosses and liverworts and those kinds of things, all the way up to corn plants and trees and whatnot, have usually several genes closely related to Siamese. So this is obviously a function that's been important in plants for a long time. But how does it actually regulate endoreplication? So let's review our hypothesis here. We think about things with a hypothesis. Well, we suspect that we know that there are cyclins. We don't know which one. We'll see in a minute there's a lot of cyclins. And we know that cyclins turn on cyclin-dependent kinases, and there are particular target proteins that get phosphorylated that lead to mitosis. And what we think is going on is that Siamese, our hypothesis is that Siamese binds to a complex like this, inhibits the activity of the CDK, and kills the activity of the cyclin-dependent kinase, the CDK, so that it can't take a phosphate from ATP and put it onto any of these hundreds or thousands of mitotic proteins, and so mitosis doesn't happen. So that's our hypothesis. But the problem is there's a bunch of different cyclins and cyclin-dependent kinases, and we don't know which one, or we're just trying to figure out which ones it binds to. Plants have 31 cyclins and 3 different kinds of CDKs, and those 3 kinds, there's actually some duplicates, so there's 5 total. That leaves a lot of combinations. We need a way of testing which cyclins and CDKs at least bind to SIM, and ultimately we'd like to show inhibition of the enzyme activity. And to test this, we use a fluorescent protein originally from a jellyfish that fluoresces when you shine the right wavelength of light on it. And in fact, this protein has been mutated so that the original one was green fluorescent protein, but humans have engineered it so that if you put the right amino acid mutations in it, it can fluoresce red, it can fluoresce yellow. I'm going to show you a control protein that fluoresces red, and then we're going to do our interaction assay with a protein that fluoresces yellow. And by the way, if any of you know, ELISA James is working in my lab, and her project actually is going to involve this technique in the next week or so. She's been building some DNA constructs to use here. So what you do is you take a gene for this yellow fluorescent protein, and if you stick that piece of DNA that encodes that gene next to the gene for your favorite protein with no stop codon in between, and put it into a cell with the right promoter and all that kind of sequence with it, the cell doesn't know any better, and it expresses a protein that runs on from one protein into the other. So at one end, you've got the piece of yellow fluorescent protein, and at the other end, you've got your favorite protein. Well, the trick we use is that you can actually take the amino terminal half of yellow fluorescent protein and fuse it to test protein A, and the carboxy terminal protein of half of yellow fluorescent protein, and fuse it to test protein B. And if these two proteins don't interact with each other, these two halves of yellow fluorescent protein are dead. They don't work. But if these two proteins actually bind to each other specifically inside the cell, now most proteins don't stick to each other. This sticking together of proteins is specific for functional interactions, at least most of the time. If these proteins stick together, these two broken halves of yellow fluorescent protein can fold back up into the full native structure of yellow fluorescent protein, and now if we shine green light on it, 514 nanometers, yellow light at 530 nanometers is emitted, and we should be able to see that yellow light in the microscope. So, and this is called bimolecular fluorescence complementation, or BIFC for short. So this is an assay to see if two proteins bind to each other. So we shoot both proteins into a cell, and the way we actually do this is we take a thin layer of onion, that thinnest layer that you can peel off an onion, that really thin layer, that's a single layer of cells. And you can lay that out on a Petri dish, and we actually take gold particles coated with DNA and blast the cells with it. The gold particles punch through the cell wall and carry the DNA into the cell, and the genes get expressed. So it's like blasting them with a shotgun, and we get DNA. It's really an air gun. And if the two proteins don't interact, no fluorescence. If the two proteins do interact, fluorescence. Well, what's this look like? Here's a control experiment where we took two plasmids, two pieces of DNA. One has a red fluorescent protein called M-cherry, and it has the full-length protein. This is a marker just to show that this onion cell took up the DNA, and its cytoplasm, including cytoplasm, there's the nucleus, but it's really the cytoplasm wrapping around the nucleus that's fluorescing. There's a big old vacuole in the center that causes that not to be so fluorescent. And you can see that that cell got transformed by the DNA. It took up the DNA, which we call transformation. And here we have a particular cyclin fused to the C-terminal half of yellow fluorescent protein, but it doesn't have a partner that it binds to, and we see no yellow fluorescence. So without a partner, this is a negative control. Okay? Without a partner, we see no fluorescence. Now, if we pair the N-terminal, the amino terminal half of Siamese, with that same C-terminal half of YFP fused to this particular cyclin D32, we see yellow fluorescence. And with a cyclin-dependent kinase fused to the C-terminal half of YFP, we also see yellow fluorescence. Does that make sense? So this is the technique that we're using. And ELISA is doing a couple of experiments. She's actually testing another cyclin that we think might be really important. And hopefully she's going to get a chance to do one more experiment that involves a different cyclin-dependent kinase. So in conclusion, Siamese is an important regulator of endo replication. I think I've convinced you of that. Siamese binds to specific cyclins and CDKs and is likely to be an inhibitor of specific cyclin CDK complexes. We want to test whether adding it to the cyclin-dependent kinases in a test tube works. I've tried that some and failed when I went on sabbatical to France last year for the spring semester. But we think we have a clue as to why that is. I don't have time to tell you. And so, but we really need to show that final step that Siamese really inhibits the CDK activity in a test tube. So there's the biochemistry, not just the tying the guy's arms behind his back and saying, oh, if we break that function, the cells divide when they're not supposed to. Well, how? We have to prove biochemically that it's really because it's affecting CDK activity. So that's the goal we're going towards. And we also need to know how Siamese itself is regulated. Now, I didn't put a slide in there, but what good is this? You know, you should always, when you do science, always think about what would I tell my mom? What would I tell my grandmom about why what I'm doing is important? I mean, I think it's cool, but you know, why would my grandmom care about leaf hairs? Well, she probably wouldn't. It turns out that one thing, I mean, yeah, this might give us a clue as to how to control mitosis in a cancer cell, but plants aren't that closely related to humans and mammals. I do honestly believe that we're getting clues as to how mitosis is controlled, and that that's going to matter in the big, big picture, okay? But closer to home, I took the slide out, but now I want to tell you about it anyway. One place where endo replication is important is, does anybody know why, in gardening, why peas and beans, legumes, are really important? Why do you grow corn or cotton on the land one year and you grow soybeans the next year? To put nutrients back in the soil. Do you know what nutrient? Nitrogen. Okay? And do you know, does the soybean do this all by itself? Does it have a partner? What's the partner? the bacterium, right? Okay. It used to be called rhizobium, and they changed its name, just like tomatoes used to be called lycopersicon, and now they're solanum. Doesn't matter. So I get confused, but there's a bacterium that does this. And this bacterium actually invades the plant cells and lives inside plant cells in nodules in the root. And it turns out that these nodules grow mitotically. They grow out of the root, almost like a side branch of the root, but they make this bulbous nodule, and then all of a sudden, mitosis switches off, and the cell switches to endoreplication, just as the bacteria are invading. And the cells become giant cells with a huge nucleus, and those are the cells the bacteria live in. And it turns out that if you short-circuit endoreplication and prevent those cells from endoreplicating, the bacteria can't invade, can't establish themselves, and the beans can't fix nitrogen. They can't capture nitrogen because it's the bacterium that does it. If the bacterium can't live in those cells, it ain't going to happen. So the dream would be if we could engineer corn plants and cotton plants so that they could make their own root nodules and fix their own nitrogen, then we wouldn't need to dump all that artificial fertilizer on the soil. We wouldn't need to alternate between beans and corn or beans and cotton, and all the artificial fertilizer we dump on when we're growing cotton or corn wouldn't flow into the Mississippi and cause algal blooms in the Gulf that causes a dead zone with not enough oxygen and kills off fish and shrimp and all that good stuff that we like down here in Louisiana. And people have shown that endoreplication is important, but nobody has looked at the role of these Siamese proteins in those root nodules. I've shown that Siamese is a key regulator of switching on this process, and I bet that bean plants use these proteins to do that. And I'd like to figure that out in the last part of my career. That's the place that I'd like to see this go someday in terms of practical stuff. Okay, I have to acknowledge a whole bunch of people. Most of this work was done by Narendra Kumar in my lab. Michelle Speckhard is the one who originally figured out the Siamese gene, along with Matt Brown. Actually, Jason Walker discovered it. I've had lots of help from the LSU microscopy facility over the years. I have international collaborators right now with the cyclin-dependent kinase stuff. Art Schnitger and some of the other people in his lab in Strasbourg, France have been very helpful, along with a bunch of other collaborators. Most of my funding comes from the National Science Foundation, and I got some money from the University of Strasbourg in France to go there in spring of 2011. One of the reasons that you might want to consider a career in academic biology is that you get to do cool things like international travel and things like that. I mean, I worked in a lab. It's not like I just, but I could still on Saturday, I could walk to the park in France, and that was kind of cool. Okay, so that reminds me that we've got the pizza party sign-up sheet over there, and my final concluding slide is that the goal of my lab is to boldly go through the black hole where no trichome has gone before. And I'll be happy to answer any questions. Questions? Yes, sir. Oh, right. Like, so the people in the distant audience can hear you.
Yes. Okay.
Right.
I work in Dr. Cotto's lab, and we use split luciferase complementation assays. How does that compare to the complementation assay that you use in your lab?
It's actually very, very similar. The split luciferase assay is in some ways more sensitive, but it's harder to actually visualize so this is, they work with a protein. Luciferase is a similar protein that emits, you can split it in half, if it's brought close, if the two halves are brought close enough together, they assemble into a functional protein, exactly like what I talked about here. Luciferase, you have to add a substrate and ATP to it, I think. And that assay is more sensitive, but I think it produces less light, so it's not as easy. It's not as easy to see where in the cell things are going on. But in fact, we're considering testing this with the split luciferase, some of our things with the split luciferase system too. Dr. Cotto is actually a friend of mine, and we share a lot of tools that we've got for working with fluorescent proteins came from him. Actually, his name was on that acknowledgement list. I didn't point it out. But yes, it's a very similar technique. Other questions? Anybody from the audience in the other places? Okay. Thank you.